Right. Now I'm going to tell same thing again because this is a bit complicated. We should a little bit slow down. We should try to understand this process very carefully. Now, what I want to show you, this is the cell membrane, plasma membrane of T helper cells. Plasma membrane of T helper cells. And from this side, I want to show you the antigen presenting cell. So antigen presenting cells, you have learned this cell membrane has a MHC protein like this. So let's say this is the MHC protein. And I told you MHC protein is very closely attached with antigen. Antigen is a polypeptide like this. This is antigen and this is MHC protein. Now T cell has a receptor. T cell has a receptor, and T cell receptor can identify this complex. So if we try to understand T cell receptor, how it looks like this. It has a shape like this attached to the cell membrane, and another one attached like this, a protein like this. So this is a T cell receptor. So I would like to make a clear point in this case. The shape is H shape. T cell receptor is H shape. So it can bind to the antigen and this MHC complex. So if it is correctly bind to the antigen and MHC complex, then there is a signal passed into the cell. At the end, it produces a protein. This protein collectively called cytokine. So we call immune system protein, signaling protein, cytokine. And this cytokine goes out, and cytokine bind again to the T cells. Cytokine bind again to the T cell. After binding cytokine to T cell, the final effect is mitosis. T cell, T helper cell mitosis. Now T helper cell mitosis also given other names. One is clonal expansion. Another name is given, same name, right? These are all are same. Then expansion and proliferation. You know, mitosis, genetically identical two cells are made. Clonal expansion, same, one cell trying to make same many cells like that. Proliferation is also same, cells start to divide and increase the number. So finally, T helper cell number increase. Specific to this antigen, right? So specific to this antigen. Specific to this antigen. That means there are other T cells, they are not specific to this antigen. When the infection is there, only that T cells specific to this antigen increasing. Otherwise, no point all antigen specific T cells are activating because they can't recognize the antigen which is infected right now. So this antigen is specific to certain pathogen. Only T cell which identify that increase the number. Right. The shape of T's receptor has H shape. I can also show it how it looks like in the real picture. You see this cell membrane with phospholipid bilayer, and you can see here attachment part to the cell membrane. This is the attachment site to the membrane, and it has another part like this, which can bind with the MHC and antigen, and there's a sulfur-sulfur bridges, disulfide bonds, and it looks like H-shaped. T-cell receptor is H-shaped. Okay. Now here you can see binding of the T-cell receptor to MHC and the antigen, same story, but I will draw it again. But I just want to focus this question. Select the correct statement regarding antibodies. We already selected the answer. They bind with specific antigen to activate the complement system. We have selected this. This is the answer. Now focus on the choice 2. They have the same Y-shaped structure as T lymphocyte antigen. That means antibodies have Y-shaped, and this is same as T lymphocyte antigen receptor. The structure is similar to T lymphocyte antigen receptor. No, I told you now it is H-shaped. T lymphocyte antigen has H-shaped. So this is incorrect, but at the end, this will increase your confusion if you go this direction again, because if you learn well in your theory and you don't miss anything, then you can remember, as I told, that T-cell receptor shape is H-shaped. And he told always antibodies are Y-shaped. Okay, antibody is Y, T-cell receptor is now H-shaped. Okay, that is wrong. For that, the knowledge of learning T-cell receptor H-shaped is important. We learn always antibodies are Y-shaped molecules. But today you learn T-cell receptor are not like antibodies shape, they are H-shaped. Right. That information is also important to learn. Why? Because you see a question. That means they are important. If there's a question, these details are very important for us. Right? So again, go back to the binding things. Now here, antigen presenting cell and MHC, and this is antigen. Now this antigen is a small, we can consider as a polypeptide chain or small protein molecule. Polypeptide chain is a amino acid sequence which is folded, not much complicated. But the protein comes, the protein has a little bit advanced structure. It is folded in a bigger way. I hope you now 3-4 times you get this information. Antigen presenting cells, it has MHC, and it has now an antigen incorporated or attached or bind to the MHC protein. Now here is a T cell, T helper cells. Now I can say antigen has several binding sites, right? This has several binding site. I write it antigen, right? Antigen has several binding site. So several binding site I can number 1, 2, 3, 4. So binding site for what? Binding site to a T cell receptor. Here is a T cell receptor. So at this case, T cell receptor can recognize this. For example, like this. So this number 4 recognized. So number 4 is now considered as an epitope. So what is epitope? Specific binding site of antigen with antibody. Specific binding site of antigen with antibody, because if you look, if you have your resource book in front of you now, and there's a choice is given, antigen has only one epitope. You ask, select the correct answer. One choice is given, antigen has only one epitope for antibody. That's wrong, because it has several epitopes. Several binding site. The binding site are called epitopes. Epitopes, again, we can define specific binding site of T helper cells or even B cells. We can take later, we haven't learned B cells, but here we take only T helper cells, antigen with antibody. Now this is the T cell receptor. T cell receptor bind with antigen, and this exact binding site is called epitope. I will tell this in B cell as well, but slightly differently when we learning B cell. Now we are learning T cells, T helper cells. Okay, this is the end of T cell, T helper cell activation. We learn a little bit more detail T helper cell activation, because immunity, if an examiner wants to make questions, the examiner know this is the most important information in immunity, activation of T helper cells. So activation is T helper cell is the main and the most powerful loading of the weapons against this pathogen. T helper cells are the main weapons loading or activating in this pathogen infection. As a result, not only A-level, when we are advancing in medicine and other lessons, this become one of the most important lesson for all the student. As a result, we should frequently expect question for our exam, T cell activation. Right, so if pathogen pathogen wants to kill the human, let's say pathogen wants to kill the human, immune system activate, immune system try to kill the pathogen, but pathogen thinking differently, I should definitely kill this person. Then pathogen can attack T helper cells. If the target is T helper cells, then immune system is completely going down. So at the moment, there are few diseases in the world. If the pathogen enter into the body, then there's no way to cure. If the pathogen enter into the body, then there's no way to cure, because one group of pathogen, they attack in T helper cells. By attacking T helper cells, destroy the immune system. I think everybody know this disease. It's acquired immune deficiency syndrome, AIDS. The virus select T helper cell as the host cell. Virus is going to the T helper cells, destroying the T helper cells. So you can understand how important T helper cells. So we learn activation of T helper cells as the first mechanism of activation of adaptive immune system, right? Okay, now this picture again shows you this is antigen, and you can see several epitopes. Epitope 1 epitope here, 1, 2, 3, 4, 5 epitopes. Antibody are specific to binding to the epitope. So antigen has several epitopes. Antibody are specific to one kind of epitopes. So similar epitopes. In this case, these two epitopes are similar. So we haven't learned antibody. We'll keep this what is antibody binding site and how it binds like things later. But this picture again give the idea. Antigen is a polypeptide chain with several binding sites for antibodies. We call it epitopes. Okay, now where we are in the adaptive immune system, I can show you like this. So adaptive immunity can be divided into two. One is cellular, other one is a humoral. Now, cellular immune response done by T lymphocyte. Humoral immune system done by the immunity done by the B cells. Humoral immunity, the part of the adaptive immunity manifest by the B cell activation. Now here, T cell activation is the main point. Now what we learned today, activation of T helper cells. Now question is, why we learn activation of T helper cells at the first place? I think answer given before, they are the most powerful cells. Now the question is, what make them more powerful? Why they are more powerful? So we're going to address this question in this lesson now. Why they are powerful? Now, T cell main function. What is the main function of T cells then? Can T cell kill the pathogen? No. Can T cell make antibodies? No. Can T cell do phagocytosis? No. So T cell can't do anything. Then why T cells are important and what is the exact function of T cell? We should clearly understand. Okay, first thing, we're going to learn what is the role of the activated T cells in this place. Here. So if a T helper cells specific to this antigen activated, we get large number of T helper cells. So first thing is adaptive immunity is to increase specific T cell helper cells for this antigen. Then their availability is high. their availability is high. When their availability is high, they can interact with other cells. Let's see how this happened now. Okay. Now here, which cell can kill another cell? Which cell can kill another cells? We learn phagocytic cells. They are the main cells. They can kill another cells. Now I'm going to introduce you another cell. Okay, we learned another cell before. NK cell. Second cell is NK cell. NK cell called natural killer cell. They can also kill another cell. Phagocytic cells can kill a cell. Second cell, natural killer cell. So it can kill the cells. Third cell is called T killer cell. It's again T lymphocyte, and it is called T killer cells. And this has another name frequently used in a modern textbook, CD8 cells. T helper cells, usually we refer as CD4 cell, but no harm of remembering this name. There's another name for CD8, CD4. So T killer cell and T helper cells. Now T killer cell can kill another cell. Why it killed the cell, how it killed the cell, and how it can get this ability to kill the cell, we're going to learn. Okay, now look carefully. Again, I'm going to do the same thing. This is the antigen presenting cell. So you have learned, I hope you remember, it has a protein called MHC. MHC attached with the antigen. This combination can be recognized by T helper cell through its receptor. Then what will happen? These cells activate and produce large number of cells. Specific to this antigen. Large number of T helper cells produced specific to this antigen. The antigen is this one. Now, T killer cell. T killer cell can also recognize same story. Same thing, antigen and MHC. And this is a T killer cell. It's different color. T killer cell. So it can also recognize same complex like T helper cell. Right? T killer cell recognize antigen. So here there is no difference now. T helper cell and T killer cells look same. They're more similar to each other. Their receptors are like this. They can do the same function. They can recognize the antigen presented by antigen presenting cell. But it cannot undergo mitosis. It cannot proliferate. It cannot do clonal expansion. It just bind and stay here. It cannot do rest of the thing. Generally, it cannot proliferate. It cannot undergo mitosis. It cannot undergo clonal expansion. Then what is the purpose of just attaching to this? attaching to this, now coming the important point. Important point is now this T helper cells, so the helper cells is activated one. Activated one mean it just know this antigen and the numbers are a lot. They have large number of T helper cells now. Now these T helper cells can give the help. So that's why it's called help. So what is the help? It gives the chemical signal using cytokines. Cytokines are chemical messengers, group of chemical messengers in all immune system cells, and they can give the message to another kind of cells. Now T helpers, activated T helper cells provide cytokine chemical signal to T killer cells. Now T killer cell can undergo mitosis. T killer cell can undergo mitosis. What is the purpose? The T killer cell specific for this antigen increase. T killer cell specific for this antigen increase in number. Because body need cells specific for the antigens at the moment. Now this is a T killer cell activation. Right. Now we have another weapon, T killer cell. So how T killer cells kill the cells? How T killer cells helping phagocytosis? We're going to learn.
Sir,
You have a question?
yes, sir. This T helper cell, it will replicate itself, no?
Yeah.
What is the use of replicating itself? You explained, I didn't hear it right.
Yes, I think if you, it's better that you use headphone or, I mean, I usually recommend if you listen, better to use headphone or earphone, because when our voice convert into electronic signals, and then it come again from your side with the speakers has vibration sometimes. The computer speakers vibrations are noisy a little bit. It make a lot of background noise. I recommend to use headphone or earphone, because if you hear well, then only you understand well. Anyway, yes, I didn't explain what is the role of T killer cell now. Yes, we try to learn T killer cells. Right now, T killer cells, we have another name, TC, cytotoxic T cells. So we can use T killer cells, cytotoxic T cells, CD8 cells. They have a different names, but it's the same cell. Refer to the same cell. Now here cytotoxic. It's a toxic to the cells. The meaning is toxic to the cells. So it eventually kills the cells, right? So we're going to learn in this case, how T cells, T CD8, T killer, cytotoxic T cells, same cells, what they can do. Okay, let's assume viral infection. If there's a viral infection, viruses are non-living things. Viruses are non-living things. Basic mechanism is they need a host cell, living host cell. So when the virus is going to the living host cell, it starts to replicate. So it uses the host cell protein synthesis mechanism, enzymes, proteins, amino acids, other resources. They're like a parasite, intracellular parasite. They're hijackers. They use all the system of this host cell and then increase number. So they increase inside the cells. Now there's a problem. The immune system cannot attack virus because they're inside the cell. That's one of the biggest problem at the first steps of immune system. They are growing inside. So it's very difficult immune system to get these cells. Let's say the phagocytic cells are coming to this place. A phagocytic cell cannot attack own cell. This is the own cell of the body. This is the own cell of the body. Phagocytic cells not attacking own cells because this is self. It's self. And that's a big problem in viral infection. They are growing inside the cell. Now, in this case, I will briefly explain to you what is going to happen. Now, this cell, the host cell, in fact, the self, that's a self, can still present small piece of antigen in the cell membrane. So some of the virus antigens can still show in the membrane like this. And it is again like before, the protein in the cell membrane, MHC protein are there. And then antigen attached to the MHC and showing in the cell membrane. So this is a sign indicating the cell is infected with virus. Now, in this case, the T killer cells, cytotoxic T cells, specifically armed to identify this combination. Now, cytotoxic T cell can recognize this antigen because this is activated cytotoxic T cell. Then next, this cell secrete enzymes. So enzymes secrete, an enzyme lyse a plasma membrane. Cytotoxic T cells secrete enzymes. Enzyme lyse the plasma membrane of the cells. Then what will happen? when the cell membrane damaged, these virus particles release out. The virus can be again phagocyted by the phagocytic cell now. The phagocytic cell then start to phagocyte the virus. In that case, T killer cells are important in viral infection. Or T cytotoxic T cells are very important because they can specifically find infected cells, and then they lyse the infected cells. Virus are released. At that time, phagocytic cells can take this virus for phagocytosis. So this is one function of cytotoxic T cell in viral infection. And there's another important role we can also see. So cytotoxic T cells, T killer cells, so CD8 cells, same cells, when they activate, they're also important in the cancer. So certain cancer cells, not all cancers, they express a specific altered proteins. So this is a cancer cell, and this is an altered protein. You know, cancer cell is own cell. They are self. They are not outside cells. Cancer cell is body's own cells, but they lost the cell regulation. They can't control the cell cycle. They can't control the mitosis, and they start to increase the number. So they try to increase the number, and many cells are produced in a place because it's not controlled by the cell cycle. Certain cases, immune system can still protect us from cancer by killing the cancer cells. So again, same thing happened. T killer cell can identify this combination, and they can kill the cells by secreting enzymes, lyse the membrane. So T killer cells are very important cells in cancer and viral infection. So they have ability to kill the cells at certain level. Cytotoxic T cells, the role is like this. Okay, next we're coming back to this place. We learn helper cells give the signal for the cytotoxic T cells, and they are important virus and cancer conditions. Next, the role of B cells. What is B cells doing?